So finally, we'll talk about nucleic acids. Nucleic acids are essentially our genetic material. So they make up our genetic information and they help to make proteins to make traits. So our nucleic acids, they actually play a direct role in protein synthesis, or the actual making and building of proteins. And remember, we need our proteins in our body because they have so many functions that help us to maintain homeostasis. The atoms that are present in nucleic acids include carbon, hydrogen, oxygen, nitrogen, and phosphorus. So all five of those essential elements that we talked about in the beginning of Unit 2. So because carbon is present, nucleic acids are considered organic molecules as well. The three polymers, or the three main examples of nucleic acids we'll talk about in this course, are ATP, adenosine triphosphate, which is our energy, or it's a usable form of energy in the body, DNA, which is deoxyribonucleic acid. That's how our genetic information is actually stored, and that can be found in the nucleus of every cell. And ribonucleic acid, which is RNA, and those molecules will help to actually build the protein. So they're actually going to move specific molecules around to help put the proteins together themselves. So the subunits or the monomer of a nucleic acid is a nucleotide. So it's kind of easy to get those two switched up since they sound so similar, but a nucleotide makes up nucleic acids. There are three parts of a nucleotide that you will need to know. A nucleotide is made up of a sugar, a phosphate group, and a nitrogenous base. There are actually 5 nitrogen bases that you need to know as well. And we'll touch back up on these later in the semester as we talk about what they mean specifically. So there's A for adenine, T for thymine, G for guanine, C for cytosine, and U for uracil. So those are the 5 nitrogen bases that could be found in a nucleic acid. So the nitrogenous base would be up here, as we can see color coded in green to the right. It would also contain a phosphate group and a sugar. This is one nucleotide. And so remember, many nucleotides together will help to actually create that polymer, such as DNA, RNA, or ATP. So typically, DNA we know is in the shape of a double helix. So as we can see in the pictures on this slide, there are two strands twisted around each other. And we can also see that there are some letters here. So the A, T, C, and G, we can see that they are matched up in the middle, these rungs here in the middle of this twisted ladder. So these letters, or the adenine, thymine, guanine, cytosine, they hold the specific genetic information that helps to control the rest of the cell and the rest of the functions. So on the outside part, we have the sugar and the phosphate. So we say that's the sugar phosphate backbone, but remember the nucleotide has a sugar and phosphate. Just make sure to make that connection there. And so essentially, our monomers, again, those are the nucleotides, which are sugar, phosphate, and a nitrogen base. Those 5 bases are A, T, C, G, and U. And polymers, or examples of nucleic acids, are DNA, RNA, and ATP. So I've mentioned DNA and RNA, so deoxyribonucleic acid and ribonucleic acid. DNA is a double helix molecule, so it's double stranded. It contains the 4 bases A, T, G, and C. The reason it's called DNA is because it contains the deoxyribose sugar. Notice that it ends in ose. Remember, ose means that it's a sugar. DNA is responsible for storing genetic information. RNA, on the other hand, is a single helix, meaning that there's one strand. Notice here that it contains the nitrogen bases AUGC. So for RNA, there is no T, it contains a U instead. While DNA does not contain a U, it contains a T. So make sure to know that difference. RNA contains the ribose sugar, so again, that word part ose means it's a sugar, and they are responsible for delivering genetic messages and helping to actually synthesize those proteins or to actually build them. So just to recap on these macromolecule chemical indicators before we get to our lab, our Benedict solution is for simple sugars. This is the only one we'll need to use the hot plate for, and if it's a positive color change, if there are simple sugars present, then it could turn green, red, or orange. When we use iodine solution, that's to test for starch, so those polysaccharides, complex sugars. If there is a positive test for it, it will turn dark purple or a black color. When we use a brown paper bag, we're testing for lipids or fats. If it is a positive test, it will leave a greasy, translucent residue on that brown paper bag, letting us know that lipids are present. And finally, that biuret solution is the test for proteins. If it is a positive test, it could turn like a pink, purplish, periwinkle color that will let us know that there are proteins present. So, just to practice with those chemical indicators, take a moment now to pause this video to try to answer this question. So, the answer should have been C, starches and lipids. The iodine test and the brown paper bag test were both positive. Iodine, we know, tests for starch, and brown paper bag tests for lipids. So, the answer would have been C.